join me in welcoming to the Distinctive Voices podium, Joan Strassmann.
I'm really humbled that so many of you decided to share your evening with me. It's really a great pleasure to be here. I left St. Louis this morning and hit Denver and all the snow, and I thought, I'll just stay here and go skiing, but no, the de-icing machines came out. Anyway, here I am. My talk. I should give a talk. I should actually give a talk, shouldn't I? I have to give a talk tomorrow, too, to a professional group that's meeting here. And this is the talk that I worked on because, if I can't tell you guys some interesting stuff, who can I really talk to? Where's the old, scientist? Okay, so, I'm not so in love with lectures myself. I wasn't that good of a student. I'd always rather hear my own ideas than other people's ideas. I've learned actually to pay attention. Anyway, when there's a red question, I would like you to just talk with your neighbors about what is the answer. There'll just be a few of these, and we'll just take a little while, and I'd just like to start that way at the beginning so you get it. So here's a picture. These are blue-footed boobies in the Galapagos, and what's going on? Just take a few minutes to either think about it, talk to your neighbors, and just decide what's the story. Okay, so maybe by now you have some idea of a story, and I just have a couple questions. I can imagine what some of your stories are about. What I'd like to know is, did your story include this guy over here? Did it include, who included that guy in their story? Oh, a few of you. How about this one right here, right behind this one? Did that one, was that one in your story? Yeah? So this is what we scientists do. We look at nature and we make up stories that try to explain it. Okay, but that doesn't quite make it science. What we try to do is then come up with some kind of general theory about what we've seen, right? It can't be just stopping at the story stage. Now, when we go to test that theory, we can't look right back at the examples that caused us to come up with the theory, because that would be circular. What we have to do is say, okay, if this is going on here and this theory is general, then when we look at another example of a similar sort of thing, then that theory should also hold. And if it doesn't, it's not really a good scientific theory. And this is how we test. This is really the foundation of all science. And, if I were a physicist or trying to land something on a comet or something, probably the stage in my career at which I had things I could show you to develop stories would be a long time ago. But I'm an animal behaviorist, and so my stories are, they're about sex, right? They're about social behavior. They're about lots of really fun things, and they're not as hard to explain from the beginning. But all science really works this way. We make observations, we make generalizations, and then we try to test them on something else, because that's the only way we can prove ourselves wrong. And a theory in science has to be falsifiable. All right, so here you get another chance looking at something. These are black-bellied whistling ducks from Brazos Bend State Park in Texas. Again, take a minute or so, or 20 seconds, and think about what's the story. And remember, I'm going to have a trick, right? Okay, so in this example, you've probably been looking really hard, and you probably saw that this one has an orange beak and that none of the others did. So you're going to probably understand that this is actually two parents and they're young. Now you might say, well, I don't see another orange beak, and that would be true because her beak is hidden. Right? But it's there, and I know it's there because of her leg colors. So scientists have to look hard at what they study, and they have to look for what's hidden, and what's going on is another part of the whole story. Well, I've spent probably most of my career actually looking at wasps and not birds. They're easier. And so here, again, take a minute or two, a few seconds, and just what are you seeing here? Okay, I would like you to just spend the rest of your lives looking hard at animals and what they're doing, because it's really a whole lot of fun. What I see here is this is the queen. I always look for these ladies right here. She's mad. Her front legs are waving. I can see that. They're off the nest, and that's like warning sign number 1. And you go too much farther down the road, and you've got yourself a sting. I see these are newly born. They have black eyes. The others don't. I see the brood in the nest. I see these black cells have already had workers hatch out of them. I see a lot in this nest. Well, you've probably figured out this isn't actually going to be a talk about animals. So what I'd like to do now is just tell you 3 really important things we've figured out from many decades of watching animals that I'm then going to apply to a microbial system. And it's going to be about cooperation. That's what I work on. My kids would probably say, yeah, you work on cooperation. All you do is argue with us. But yeah. So sort of the first rule of cooperation is that there has to be an advantage to cooperating. Something 2 do better than 1, than each alone. So I'll just call this 1 + 1 = 5. And you may think that sounds really improbable, but say that you're a flicker and this is your nest and you're a single parent. Every time you fly out to get food, your babies are vulnerable. If instead you have a partner, you can have someone guarding that nest 100% of the time. So you can see that is really an advantage that doesn't go from 1 to 2, but way much farther up. So we always, if there's social behavior, we want to see a real advantage to it that's not just the sum of its part. Another thing we know about social behavior and cooperation is that an awful lot of it has to do with families. Individuals help their relatives. You can have genes that select for altruistic helping that never get a payback to yourself because you have passed on genes through a relative. Of course, every parent knows this, but it also applies to other kinds of relatives. So this is the second really major point. Then the third is, of course, there is helping and social behaviors among non-relatives or even members of different species. Here we have a big grouper who's being cleaned by cleaner fish. So these are sort of the 3 principles of social behavior that an awful lot of animal studies have shown. We can see these things in animals. We can see what's going on. We can formulate hypotheses and move ahead. But as I said, to test our theories, we need to test them in something different. Now, sometimes the thing we test them in doesn't have to be real different if they don't share the trait that we're looking at. So here, these are 2 kinds of wasps. These are really mean Venezuelan wasp. We ran into a fair amount. Stinging wouldn't be a trait you could use these to compare because they both have it, but there's other things that you could. But how about if you go for social cooperation really far away to something that doesn't have a brain, doesn't have cognition, has no shared evolutionary history of social traits. And so about 25 years into my research career, we made this huge switch from social wasps to these social amoebae. And I can tell you that's quite a story right there, is the insanity of making such a big shift. Yeah, I stayed married to the same guy, though, so it was fine. All right, so what I'm going to really spend most of my time on is social amoebae. Any of you heard of social amoebae? Does it sound like an oxymoron almost? Well, this is a social amoeba fruiting body. This is the stalk. Those are cells that have died. These are spores up here, and they've lived. So this is sort of a social insect colony all in one go, where the stalk cells used to be free amoebae. They die to help the others. So that's your social story right there. You can get at genes. You can do lots of things with this system. It's been really, really fun. All right, so what are they? They're in the amoebozoa. This is sister to animals plus fungi. Bet you didn't know fungi were more closely related to us than they are to plants. I sometimes try to tease vegetarians into not eating fungi because they're so close to us. Anyway, the common ancestors were a really long time ago, and we didn't even have this much oxygen in the air. It's a cool place to be on the tree of life. Social amoebae are everywhere. They make great little kitty projects because any soil will have them. There's about 200 species. They are eukaryotes. They are not bacteria or protozoans. They're eukaryotes. They have a nucleus. They have mitochondria. They're really kind of just like us in terms of a lot of bacterial diseases. So they're used as models. They're haploid. So we're diploid. We have 2 copies of each of our chromosomes. They're haploid except for during the short sexual stage. They're hunters. They eat bacteria. And they are amoebae, so they divide by binary fission. Okay, I know none of you were expecting a lion talk. I'm pretty sure of that. But if you were, and you're feeling a little disappointed right now, maybe you could just think of this as a lion and this is a gazelle. See? How cool was that? So here's a amoeba doing what they do, divide by binary fission. This is one of their ways of dividing. Okay, now, I said there's about 200 species. I'm actually only going to talk about one of those species, and that's Dictyostelium discoideum. And I did, after a while, learn how to actually say it correctly and even spell it correctly. Mostly today I'll be calling it Dicty. People work on it more than others. It's become a huge model system for all kinds of cell biology. It has this slug stage where it doesn't leave a huge tail, and that makes it a lot easier to study. This is the point I would like to tell you about my dear husband, who is actually enjoying a very nice little reception with lots of wine at the hotel we're staying in. We work together. We have a great partnership. We have both the same and different strengths. Here he is collecting soil that has Dictyostelium discoideum dicty in it from the place where it was first collected in near Mount Mitchell in Virginia or North Carolina. Sorry. All right. So what else? So that social stage with altruism happens when they starve, and there's all kinds of things known about the signaling and stuff. and then they make that stalk, and I'll show you some of those processes. They also have a different social stage, which we call sex, and it's really interesting. It involves what I'd call extreme sociality and cannibalism. All right, so this is a pipetman tip on the left, releasing cyclic AMP, which is one of their main attractant compounds, and there you can see the amoebae really love it. They're just going towards it. They smell it. They love it. They are attracted to it. It's really an interesting compound because it is an important second messenger inside cells, but here it's used outside cells. So they chain in, and then they form this. This is at a lower magnification, and they're just swarming in, and then they make that slug stage, which I showed you earlier. And then ultimately they make the fruiting body, where some cells die and others live. Now, it's nice to just sort of think about these as sort of like an animal when they're in the slug stage. So this video made by one of my grad students, who was actually my major professor's son. So yeah, it was in the South, but anyway, it's a little bit interesting. He's a great student. So just watch this. Is this what happens to you when you walk by friends? Do you merge? So the green and the dark and the unlabeled are two different clones. And so sometimes they're separate, but then they just kind of get sucked in. Isn't this mesmerizing? It's just like, this is speeded up, of course, but it's just so interesting to think about all the different kinds of processes that are going on there. All right, but I'm going to tell you a fairly simple story that just makes these 3 points. So we can then ask, what is it that dicty does that's social? And they really do these 2 things. They aggregate and move, and they form a fruiting body, which I showed you, in which some cells die. They move farther if they're in groups. So the individual amoebae can only move very short distances, whereas when they form this multicellular body, they can move farther, and bigger groups move farther than smaller ones. Well, this is a cartoon, and maybe if I don't show real data, you'll think I'm not actually a scientist, which probably I would take as a compliment, because it might mean I was communicating. Anyway, so here's some data from Kevin Foster, where this is slug size in terms of volumes of spores and distance traveled, and so you can see that the bigger they are, the farther they travel. You heard I was president of the Animal Behavior Society. Given all the stuff I've done on WASP, that probably didn't surprise you, but what's also true is that I was elected president of the Animal Behavior Society 10 years into working on these things, and it was really, they're not animals, and they hardly have behavior. But there is this one behavior that they do have, which is they will move towards light in the social stage. So we have done so many experiments racing them towards light. So when I go to the behavior meetings, I have that little tiny piece to talk about. So here's an experiment where we took slugs, we raced them towards light across bacteria to see if they'd starved to be slugs, could they be like, oh, never mind, it's Thanksgiving, or not. So this is, I will try to show generally the first author of papers, Jenny Kudzel-Fick, who just had a baby 2 weeks ago, raced these slugs. And what she found is they went right across the food, but they seeded it so that they'd left babies behind. They'd left cells behind, so they were able to use it. So that's an advantage to moving over distance. Another advantage, we hope if they're going to, 20% of them are dying, that there is an advantage to being lifted up. One of them could be they're getting away from the soil. You may notice this isn't actually soil. This is a dead fly, which landed in the plate, and then bacteria found it, and it became a great place to eat. So Jeff Smith wanted to see, well, do they actually get picked up, and are spores more likely to get picked up with stalks? So he did a somewhat artificial experiment using fruit flies and either knocking down the stalks so the spores were on the ground or not, and then turn them on their side so it wouldn't just be gravity, and found that the intact fruiting bodies were actually carried by flies more than the disrupted ones. So these are the ways that 1 + 1 = 5. Groups move farther, they leave sprouts, and the fruiting bodies facilitate dispersal. So this is the kind of first thing you have to know is there any point? Is this really social behavior? All right, so yes, there's an advantage, but I'm sure you know that in many kinds of social circumstances, everybody may benefit, but some people or some organisms benefit more than others. For example, in a school of fish, they may all benefit a protection from predators, but if you're in the middle, you'll benefit more. In families, also, in social insects, you may help the queen to whom you're closely related, but of course it's better to be the queen. So who gets the advantage is also important to understand. And in the social amoebae, the cell biologist had figured out pretty early on that the front cells corresponding here to the front become stalk, not spore. So these are the ones that are paying the price. They're the kind of drivers saying where they should go. They also pay the price of becoming stalk. They tend to be weaker cells. Maybe they've divided more recently or something like that. So I won't show you data for the first point, but this is well supported. For the second point, we thought, you'd think the first to starve would be altruist, because who would pay attention to a selfish cell, right? I'm starving, y'all come over here and help me out. By the way, you pay the price. Didn't seem likely. Again, Jenny did this work. It actually turned out that when we starved cells for 4 hours, more of them became spore than the others. So that really surprised us, that the first to starve were more selfish than the later joiners. Okay, so here's another question. It's getting kind of boring, isn't it? I'm doing all the talking. Really bad. Very impolite. So if some amoebae die to help others, is this likely to be a story about partners or about families? Just talk among yourselves for a few seconds and we'll vote. Okay, how many think it's a story about partners? How many think it's a story about families? It's a smart crowd. If some are dying to help others, it is usually going to be a story about families. If it's about partners, each of them is going to be more likely to have to benefit. Okay, so if families are important, how on earth does an amoeba that doesn't even have a brain or a single neuron know who's in his family or her family or its family, right? Well, it turns out that it's hard to think like an amoeba. I thought it was hard to think like a wasp, but that just has nothing on thinking like an amoeba. But we do know that how they're spaced in the environment can affect relatedness, because if one spore lands somewhere and then divides and divides again, you may get plaques like this where all of these come from a single spore. And we can actually quantify that, and this is Jeff Smith's work, where the y-axis here is relatedness, and just to remind you, 1 would be full identical twins, 0.5 is siblings. So if you have 100 spores on a standard size Petri plate, that gives you quite a high relatedness. So just by the spatial distribution, it's likely to be pretty high. But of course, this is a Petri plate, and last time I checked, there weren't any Petri plates in the wild. So we did a transect where we filched some straws from Subway. There was one on campus. And sampled the soil with just pairs of these straws. We picked up in each one just about 1/5 of a gram, so really quite a small amount. And then we plated out the soil, collected the dictys that were in it, and then genotyped them using DNA markers to see how many different clones we got. So the blue is a different clone. So sample 1A had 2, 1B had none. This is Angelo Fortunato's work. We had this great, so just to tell you what kind of scientist I am, we were working on wasps in Tuscany. Yeah. We had 6 Italian grad students. We had lots of friends. We had more dinner. Anyway, yeah. We switched to slime molds in Texas. So isolates we could tell simply by plaques on the plate, how many clones we had to plate them out. So you can see that in any given sample, there were between 1 and 6, I think, different clones. So I thought, okay, maybe that's good in the 1/5 of a gram. Even in a 1/5 of a gram, 6 clones might actually never run into each other. So what we really wanted to know is how many were in the fruiting bodies. And we were told, so there's this annual meeting, there's a listserv, the dicty people are really friendly, but they're almost all cell biologists. And so they thought we were kind of an oddity. And we were told that we would not be able to ever find wild fruiting bodies and that even some luminaries in the field had not. But, you know, we're not dumb, and we know where there's bacteria. So we went to Mountain Lake Biological Station to hunt for them. And this is my husband, Dave. This is Angelo. This guy who looks like he's about to eat. This is a pile of deer turds. So anyway, this is Rich Kesson, cell biologist, really great guy. At the time, he was associate dean of the med school at Columbia, kind of a busy guy. But he made time. He could not believe we were going to do this, and he thought we might succeed. So he invited himself on the trip and came along. And sure enough, the first pile of deer turds that we looked at had fruiting bodies on them. So they were kind of like aged deer turds, you know, so there'd been bacteria there, and then it had been eaten. And so then we were able to genotype all the clones in those fruiting bodies and get at relatedness. This is Owen Gilbert who did this. Here's relatedness, 0.86, so that's closer than full sisters. So in nature, there's quite a bit of sorting. It could be because of how well they're distributed, but we also thought they might actually bump into others. So at this point, we had started working with a couple of molecular biologists at Baylor College of Medicine, a couple of really great guys, really brilliant, Adam Kuspa and Gadi Shulsky, and one of their grad students, Rocio Benaventos. And they just kind of intuited that maybe this gene that they already knew about, which was a cell adhesion gene, which at the time they were calling LAG, but once they realized how cool it was, they changed its name to Tiger. And, you know, for species names, there's all these rules that you have to follow, and you can't, and there's committees, and there's like, you know, you can't just do it. Apparently with genes, you can just change them. So they decided that this gene was it. We had some preliminary data. They then did something which actually took them 3 years. Drove me crazy. Nothing I do takes 3 years except raising kids, and that takes like 3 decades. So they took these genes and replaced them in a uniform background. So C1 and B1, it's a receptor and it's, lock and key sort of thing. And this video, the control is over here on the left, and this is the experimental, and watch this video. It's only about 5% of the cells are labeled is why you can see it. Isn't that awesome? So this is their work, really, really amazing results, that there's really a lot of sorting at this stage. This sorting doesn't stay perfect all the way to the end. You remember that earlier video, and I bugged them a lot about why they didn't show the fruiting bodies. But clearly there's a kind of recognition going on, and these 2 cell adhesion genes are involved. Okay, so this is the cell biology approach to this. But as evolutionary biologists, we know that if these are really the genes, they need to be really variable. They need to be under something called balancing selection. So here's some other genes just to show you what's normal. So what you're looking at here is a couple of genes. This is just reading along the gene, along the chromosome, base after base after base. The y-axes are just indicators of being different. Do you have an A there instead of a T, etc. And you can see that here and there's a little blip of difference among these different strains, but not much. This is what the tiger genes that are actually active in recognition look like. Tons of variability at these loci. So the evolutionary approach also makes it look like these genes are what's important in recognition. So in nature, if they do run into a different clone, which they're not that likely to do, but if they do, their tiger genes can help them exclude the other type. And this is sort of a two-system situation, and these are actually really common in biology. There's an awful lot of functions that have backup functions, that have redundant capabilities. And I know that initially when cell biologists started being able to knock out genes, and the commonest result was they'd knock out a gene and nothing happened. It was so frustrating because they knew that gene was involved in the process. The problem was they didn't know all the other genes that were also involved. All right, so at this point you might think, okay, this is kind of boring. They never really run into strangers. If they do, they exclude them with their tiger genes. So where's the story of cooperation and conflict? Well, one thing about evolution is it's had a really long time to act, and even just a little bit of mixing can create powerful selective forces. So remember, 20% of those fruiting bodies are dying to lift up the others. We've done lots of different, really fascinating ways of looking at that. We've done experimental evolution. We've done lots of things, but I just had to pick a few stories, unless we want to have an all-nighter, do we? All right, so if we hold, remember I said before that larger groups move farther. If we hold group size constant and mix two different clones, they move less far. It's kind of like they're fighting. You be first. You go to the front and die and be a stalk. No, you. So anyway, so they're dragging behind. And again, here are the data. Distance that a single clone goes, a mixture of 2, and then 5 or 10. So does this really mean that there's conflict with one clone winning out over another, or is it just a dysfunction? So this is work done by Neil Buttery, not with our lab, where here the X-axis is the fraction of cell input of clone C. So 50%, just try different ratios. And then the y-axis is just the output. So the neutral expectation would be this dotted line. In this case, clone C gets way more spores than it should have by rights. And this is a kind of experiment we also tried to do. We didn't do it nearly as well as Neil did. And one of the things that happens in science is sometimes people get jealous, and it's really bad. It's not good for science. It's great that he did this work, and then it lets us do other things. So I was, busy suppressing all those jealous thoughts, and welcoming the more generous thoughts was made easier when I realized he did this as a grad student. So I hired him. So that was the solution to that little problem. He's a great guy, did great work. He's now actually left science, and he has two fantastic food blogs. He's English, and he loves English food, and he cooks it so perfectly. And he runs pop-up restaurants and farmers market stuff in Manchester. So Neil Buttery. His blogs are, one is Neil Cooks Grigson, which is Jane Grigson. It's a classic English food book. The other is British Food, A History. He's really a great guy. We also found, actually earlier than Neil's really nice work, that among these clones, who cheated whom actually formed a linear dominance hierarchy. This is just the percent in different clones, and it's a linear hierarchy. So that's, we thought, was pretty cool. All right, so I've kind of shown you that Dictee cooperates with clone mates, but when they're with strangers, they do have ways of competing, and there is real social behavior going on. All right, so now I'd just like you to, I've kind of thrown you a whole lot of different things, and I'd just like to see if you can share with your neighbors which ones you've remembered and what you think is cool from what I've said so far. Okay, let's see how many of these you got. So you notice how I'm not using an outline? Don't you hate outlines? I just feel like outlines are just like, it's like telling who did it. I mean, I just like, if you're going to tell a story, why tell the conclusion? Anyway, these are some of the things I've tried to share with you. Dictee is a eukaryote. It's sister group to animals plus fungi. They're also called social amoebae, and they used to be called cellular slime molds. They are related to the acellular slime molds, which is a whole nother crazy story. When they starve, they aggregate into a slug. Bigger slugs move further. Some cells die to form stalk, which helps disperse the spores at the top. Wild fruiting bodies are highly related. There's tiger genes. Chimeras move less far. Some cheat other clones. So here's a few of the things I've shown you. All right. So now I'm going to move on to the last thing. And this stuff is just so absolutely, amazingly cool and crazy. It's just really recent stuff that we've been doing. And it's this last point, this last big point about partners. All right, so I'll remind you of the colony cycle, the social cycle. These are amoebae eating bacteria. Of course, there's way more bacteria, really. When they starve, they aggregate and form the slug, and then the fruiting body, and then spores which hatch. So that's the cycle. Now, normally, this cycle, they eat the bacteria, they take them into their phagosome, they eat it, then that fuses with a lysosome, which puts all their digestive juices out. And so that's the normal process. But what we discovered was that some of these wild clones actually carry bacteria with them all through that social stage. And again, this was something the cell biologist told us that, we were just showing poor technique, could not be true. So I'm going to tell you this story starting with an analogy, and this one was really quite fun to develop because I did it with Julio, who is my son-in-law. He's Peruvian. He got his PhD at Texas, which was where my daughter met him, and he's a geographer. He's actually back in Peru right now working for the UN and trying to help them figure out how climate change is impacting the food supply in Peru. Big problem, because the glaciers are melting. Anyway, so he studied alpaca herding in the high southern Andes, and he did a lot of work with that. They herd them, they bring them to different fields. They're herded in small groups that share the resources. And in a way, you could say there's 4 attributes that are essential for this kind of farming. So here I'm considering the crop to really be these animals, not plants, because it's a more direct analogy. So first, they're domesticated. They're not totally wild. The Quechua don't just eat their alpaca and harvest their fur. They let them grow and reproduce and cull them in a sustainable way. They defend them against invaders, and they have dogs that do this. And I told Julio I really wanted a picture of one of those dogs, and he said, "Are you kidding me? I could not get that close to one of those dogs," because he was actually viewed as a complete foreign outsider because he was from the big city and not from the high Andes, and then pass it on to your progeny. So those are the things I want to show you if these social amoebae actually have agriculture, and we'll just go through them. That is kind of like an outline, isn't it? I lied. All right, so first, farmers should do better, right? If you carry your own food, it should be more delicious than whatever's out there. If I go eat on the lawn and I bring my lunch and my friend just has to eat the grass, who's going to be happier? So Debbie Brock, who's a postdoc, really our lab head, she's wonderful. She just took some soil that had bacteria, wasn't treated, just from outside, and put farmers or non-farmers on it. And she was able to show that the non-farmers did not proliferate nearly as well as the farmers who had brought their own food along. So these farmers, they're carrying the food along in their spores. The spores hatch, they release the bacteria proliferate in the soil, the dicty eats them, it self-proliferates, and then moves on. Okay, but I didn't just say eat better, I said domesticate. And so just not too long ago, in a really wonderful collaboration with a group at Harvard, we discovered that there actually was a real domestication where this clone had evolved from that clone by losing essentially all of its defensive compounds with one mutation. It was a stop codon in a major transcription factor, if that language means anything to you. Anyway, it was a big mutation. It probably means these two can only live together. So this one is different. This one is not edible. It's carried. It's actually one of those dogs. I'll get to that later. So it's actually domesticated in the dicty context, this bacterium. So that was really exciting for us. They should be prudent. How on earth can you be prudent if you don't even have a brain? I mean, is this saying that these dicty would pass that marshmallow test? Where the kid has to, the kids that are successful didn't eat the marshmallows, whatever. Actually, they are prudent. So here you can see it on a plate where these are fruiting bodies. You see no bacteria. Here's fruiting bodies of a farmer, a whole bacteria. halo around them. If you look at it numerically, on day 1, these are just bacteria, non-farmers, farmers. By day 7, the non-farmers have eaten nearly all the bacteria up, whereas the farmers left about half the bacteria on the plate, so they're showing prudence. And then defense. There's a lot of bacteria that we've isolated from the dicty fruiting bodies that are not good food, and we're sort of working through them and looking at them as defense. We've done quite a few experiments showing that. I told you already about this one that's food. This one that is not food, that they cannot grow on, is actually a great defensive compound. This is Debbie with Pierre Stallforth. They're both postdocs. They were working together on this problem. And so what Pierre discovered was this inedible PF2 makes 2 compounds called pyrrolnitrin and chromine. The other doesn't mate. Here are the 2 compounds. It's been a while since I had organic chemistry, I have to say, but there's chlorines, there's various groups here that kind of look bioactive. One of these is actually a known fungicide. The other one is not known, but acts as quite a potent fungicide. So this is actually a great system for possible drug discovery. We also found that both of these diminish non-farmer growth and augment farmers. So this is just concentrations of chromine and how it affects the dicty. This is the non-farmers, it hurts, and the farmers, it helps. So that sort of supports this view of this. So then the final thing is whether they transmit it. Well, actually, this was the first thing we saw, was that in the fruiting bodies, and we've also seen it inside the spores, that they do carry the bacteria along with them from one generation to the next. So we were pretty happy about this. So up to this point, I've really told this farming story only from the point of view of the dicty, but I said early on that for mutualism, you really have to have advantages to each side. So why don't you just take 20 seconds to see what you think might be the advantage to bacteria. Okay, so here's some of the advantages that we think are likely. Transport, protection, and those sorts of things are probably what we think is going on, but we have not done research on this. So at this point, this is our story. It's not yet our science. We think this kind of agriculture is really only possible with sociality, because to have different clones protecting, showing prudence, and those sorts of things. So the other animals that do this are termites that farm fungus, there's ants that farm fungus, there's others also that I won't go into. So I've basically taken 3 stories from animals and shown you how they apply to a social microbe in ways that I hope are either convincing or entertaining, maybe the latter more than the former. I've taken theories from wasps, largely originated by Bill Hamilton, like kin selection, and then supported them in a really different organism. It's been really fun doing something really different, and I hope this has been interesting to you. Thank you.